Welcome everyone, Walt Schoenborn, Director of Digital Media here at scienceboard.net, and I'm very pleased to be here with Tim Kelly, CEO of RoosterBio. We're here at ISCT 2022 in San Francisco. And Tim, I want to first of all say thank you to RoosterBio and thank you for sitting down with us today. I really appreciate it.
Oh, thank you very much. I appreciate the opportunity.
Yeah, I want to get right into it because one of the things that I find fascinating is RoosterBio, when you guys say you're industrializing the regenerative medicine supply chain, what exactly do you mean by that?
Yeah, so I think the key thing is that it's more than just having GMP materials available, which of course we do have, GMP grade cells and media. But really what we believe we've done is to sort of, reimagine and redesign the way that MSC-based products can be produced for therapeutic use. So that means that, we've got multiple donors for different tissue types that are, fully off the shelf available in scalable formats, so different numbers of cells per vial. That's paired with specialized bioprocess media that's really tailored for the expansion of the MSCs. And then, so that's the products, but those are supported then by the DMFs, the regulatory information that's on file with FDA that really provides for, a lot of our customers provides a significant amount of the CMC section for their regulatory filing can be achieved simply by referencing our DMFs. And then the last part of that I think is worth noting is that we've also got development grade materials which are really kind of paired to the GMP. So you can translate, these are obviously much more accessible from a cost point of view, and then you can test our materials and then readily translate to GMP by leveraging those development grade materials.
Yeah, and, you guys have been on the move, the last several years, and just last week you had announced a partnership with ShiftBio, right. And I was really curious to see what are you most excited about that collaboration?
Yeah, I'm really excited about that relationship with ShiftBio. So ShiftBio is a South Korean company that specializes in exosome technology. Yeah. So they have their own IP around sort of how a gene-engineered exosome is actually produced. And the really cool thing about it from the RoosterBio point of view is that they're really leveraging all the elements of our portfolio. Yeah. So they're utilizing our genetic engineering medium to transduce our working cell banks with their lentiviral vector. They're using our cell expansion medium to then grow that stable pool to cell numbers that are relevant for exosome production. And then they're using our exosome collection medium to actually promote the secretion of those exosomes and collection. And then they've hired us to develop that process from end to end. So from the lentiviral transduction all the way through the bulk fill of the exosome drug substance. And then furthermore, we have a sort of a CDMO manufacturer partner network. Yeah. And so they'll be selecting their CMO for their clinical trial material from within that network of providers that we've established.
That's super. That's really cool. It's one of the things that is always fascinates me in this modality of cell and gene therapy and more is the collaborations. And obviously, great that we're all meeting in person again and not virtually, but to have those collaborations to actually be moving things along from that standpoint. I guess that kind of leads into my next question. What changes do you see and what changes do you anticipate, both as CEO of Rooster, but generally in the sector for MSC and exosome space in particular?
Yeah. Yeah, so I think the next few years are positioned to be really, really exciting, obviously, and throughout the CGT space. But I think with exosome-based therapies in particular, yes. I think you see it at the show here this week, and ISEV is happening in a couple of weeks in Lyon, where there's just a tremendous amount of interest and excitement about the potential of exosome therapies. And we believe that MSCs are really the optimal cellular source for exosomes because you're getting all of the therapeutic benefits of the MSC contained within that extracellular vesicle. Really, this is how MSCs exert their therapeutic benefit, is they migrate to the source of damaged tissue, and they secrete exosomes to communicate with those tissues and promote healing and regeneration. So not only are you getting sort of the drug delivery capabilities of the exosome, and you can engineer the exosome however you wish, to deliver a specific payload, but you're in essence sort of supporting the health of those tissues, while you're delivering the drug through all of this, kind of growth factors and cytokines and microRNAs contained within the MSC exosome. And that is unique for MSCs, right? If you're making exosomes with HEKs or with other cell types, you're not getting that same benefit. So that's one element. I think the other really key thing that we expect to happen, we're anxiously awaiting, as is I think most folks in our field, is the FDA approval of the first MSC-based therapy. Right. So obviously there's a dozen or so of those products approved outside of the US, but FDA has not yet approved an MSC-based therapeutic. Hopefully that'll change in the next year, and I think that'll be a watershed event.
It will be a watershed. Well. It absolutely will be. So in regards to that, and saying, if in anticipation that we should get to that next step, what are some of the things that you're most excited about? I know that we talked about ShiftBio a little bit, but for RoosterBio itself, what are some of the things that you're excited about next 3-5 years, pipeline, product, service-wise?
Absolutely. So I think, if to the extent that folks know about RoosterBio, they know about, our MSC products, our media products, and our ability to, we think better than anyone else in the world, expand MSCs to sort of clinically relevant numbers, by combining our cells, our media solutions, and our bioprocess capabilities. Yeah. So all of the, whether that's 2-dimensional or 3-dimensional, we have the conditions to expand MSCs, to numbers, to the billions and billions that are necessary to treat patients. What we're really excited about now is expanding that technology platform sort of in both directions. So, upstream of the expansion, we're working to add different ways of genetically modifying, our cells. Yeah. In some cases, that's to produce engineered cells. In many cases, to produce engineered exosomes. Right. So we already have the lentiviral transduction medium that I referenced with ShiftBio. We're also collaborating with some partners to develop additional ways to gene engineer our cells, through both viral and non-viral means. So really have a variety of ways to engineer our cells so that we can be as flexible for our client and customer needs as possible. And then downstream of the MSC expansion, we're really focused on the exosome, the rest of the process for producing exosomes, all of the downstream purification. Analytical characterization, I think, is a huge unmet need in this space. There's been some talks about it here this week. We're using, honestly, very blunt instruments to analyze exosomes today. So, we're really excited about being part of how the industry moves forward and really starts to understand and brings exosomes into the realm of the well-characterized biologic that certainly not today.
Well, you guys are doing obviously a ton of movement, both in MSCs and exosomes, as we talked about. There's I'm very excited to be able to share this information. We have a lot of people in our audience that are very into MSCs, very into exosomes, very into bioprocessing, very into CGT in general. And I appreciate your time today. Thank you very much for sitting down. I know you got a ton going on, but I appreciate you sitting down for us.
Certainly my pleasure. I appreciate the opportunity, and certainly looking forward to the rest of the conference and to where the field goes in the years to come.
We'll be hearing a lot more from RoosterBio in the very near future as well. So thanks everyone for tuning in. We'll see you soon.